Okay, good morning, everybody. I'm Stephen Isaacs, and I'm the CEO of Aduro BioTech, and I'm here to tell you today about clinical progress in cancer immunotherapy. And Aduro is an immunotherapy company, and I want to focus on our results in pancreatic and non-small cell lung cancer. So we're a Berkeley-based company. We develop by bioengineering Listeria monocytogenes to make it safe for administration to humans and effective for stimulating both an innate and an adaptive immune response. That's how we treat cancer, and that's how we address infectious disease. We've run 3 phase 1 clinical trials, and we have some very positive survival data, both in pancreatic cancer and non-small cell lung cancer. We want to do a follow-on phase 2 clinical trial that will be sort of grounded at Johns Hopkins, and there will be several other sites. And we want to do a follow-on phase 1b transfer trial in non-small cell lung cancer, and we're here looking for the funding to do those 2 trials. So why do we want to work in those spaces? Well, there are significant problems. There's about 42,000 people who get pancreatic cancer in the US every year. It's a big market, about 1.2 billion. Abysmal survival rate of less than 5% at 5 years. The non-small cell lung cancer is a similar but bigger story. About 215,000 patients a year get cancer, get non-small cell lung cancer. It's about a $7 billion market and 3.5% 5-year survival. So our opportunity is to combine our Listeria monocytogenes technology with either GVAX or with radiation to treat these diseases, and we have some very provocative data in both those indications. So our technology has been published in about 20 different publications. We were a lead story in Nature Medicine cover story. But basically what we do is we take the bacterium, we take out virulence genes, we make it avirulent, it's suitable for administration intravenously to humans, and then we put in antigens, and it can either be on the oncology side to stimulate an adaptive cell-mediated immune response to cancer, or we can do it for infectious disease as well. So this is the only real science slide, and this is showing both a liver and a lung model. On the upper left-hand side under control with those red blobs, those are liver tumors in mice. And basically, we can stimulate an innate response with our bacteria, which we take these 2 virulence genes out of, and reverse those tumors at the bottom left-hand panel. The reason that works is because the bacteria homes to the liver. In the lung, that's not true, so we do use an antigen. So we not only make the bacterium safe for administration, but we use an antigen, and on the bottom panel, it shows a reversal of those tumors. So I want to talk about the phase 1 trial that we call the CRS-207 trial. So this is a trial where we used Listeria, where we inserted a mesothelin antigen. Mesothelin is a tumor antigen that's overexpressed in pancreatic, mesothelioma, non-small cell lung cancer, and ovarian cancer. And so we used the bug that contained that antigen to stimulate both an innate and an adaptive immune response to address the cancers. The adverse events from the trial were generally mild and transient. Basically, everybody gets a fever, everybody gets chills, liver function tests go up a bit, but it all reverses within 24 to 48 hours. And the surprising thing was that in these trials, 6 of the 17 end-stage cancer patients, so these are people who have an expected survival of 3 to 5 months, live greater than 15 months, and 4 of those patients are still alive. So that's what generated the excitement and the focus that we're working on now. So really what we're doing is we're getting a synergy between 2 different vaccines, or vaccine and radiation. And this is well known in the literature. We've published on it. It's basically taking one carrier in, like GVAX, that brings in the mesothelin antigen, and then following it with Listeria that we specifically designed to bring in the antigen a second time. And in this prime boost regimen, very clearly in animal models you get improved efficacy, and now that's what we're seeing in the human setting. So combinations were very effective in our trials. In the 6 long-term survivors, 3 out of 3 pancreatic cancer patients who received prior treatment with GVAX had a 17-month mean survival versus 5 months for those who did not. 2 out of 2 non-small cell lung cancer patients treated with Listeria followed by radiation are still alive today unexpectedly, and 1 mesothelioma patient who had a prior treatment with an adenovirus containing able to express interferon beta. So basically this all makes sense scientifically. The prime boost data holds together, and Listeria has been shown to potentiate other immunotherapies, and immunotherapy in general has been shown to be potentiated by radiation. So the science story sticks together very nicely. We've designed 2 new trials, a phase 2A trial in pancreatic cancer. This is a 72-patient trial at 2 different doses. Basically, we will look at survival as the endpoint, and unfortunately in pancreatic cancer it is so quick, in terms of its lethality, it allows this trial to be done in a very quick period of time. So with funding, we'll start the trial in 2011. We do believe we will have results in 2012. We're talking to FDA next month, the Recombinant Advisory Committee, and pending funding, we can get the trial going. We also have a plan for a phase 1B lung cancer trial. This will be done at a single site in Hadassah, Israel, and it's basically the same story. We can start it mid-next year and have significant results by mid-2012. Competitively, we feel we're very well positioned. Basically, there are new chemotherapy regimens, but they're really recombinations of older drugs, and it's kind of up against the wall in terms of toxicity. And there is one other vaccine approach, but it's only in the resected setting. We're in the metastatic setting, and so we think we're very well positioned there. We have a lot of important IP, 2 patents issued this year, 2 more will issue by the end of the year, and we have licensing arrangements for the other technology that we need to bring in. Manufacturing, we've manufactured 7 lots under GMP, cleared by the FDA, taken these into 3 clinical trials, 40,000 doses for about $250,000 because we're making a bug and not a recombinant product. So our strategy really is to leverage the results that we have in pancreatic and non-small cell lung cancer and focus on those and keep those moving forward. But this is a platform. I liken the bacterium to like a DVD player and the antigens to different DVDs that we can put in on the infectious disease side and on the cancer side. And we have been able to raise about $20 million in federal contract and grant funding to support some of these earlier activities. So the investor liquidity windows are really around the results coming out in 2012 for the pancreas trial and the lung cancer trial. And finally, I'm trying to raise $12 million to support these 2 trials, and that'll be supplemented by about $3 million, which I actually think will be significantly more in non-dilutive funding. So that's the contact information, and I'll be around the rest of the day. Thank you.